2-to-1 private investment compared to public plus nonprofit. Very, very compressed period of going from a field that was a pure public works project to being something that was predominantly a private sector enterprise. So that's a backdrop. Now here's a little bit more detail on that. If you track the public value, the market value of those publicly traded companies, what you see is kind of a wild ride here. You see that there's virtually zero in 1990, and you see this huge peak, $90 billion in market value in September of 2000, about 3 months after the announcement of the reference sequence in the White House and in London. And then suddenly, a factor of 5 drop in the value of those companies over a very short time. The bubble burst, right? This was the genomics and internet bubble bursting. So the public value of those stocks dropped precipitously from 2001 into 2002, and then it began to recover a bit. This is the number of companies that went from basically zero in the late 1980s to the, I think by the end of this period it's 88 companies, although it's not exactly the same 88. We kind of reach steady state, and some of them die and some of them are born each year. And if you look at where are those companies based, these are the publicly traded firms. It's a very similar pattern, but the companies, the number 2 and number 3 change, whether it's public companies or private. But roughly 2/3 of this commercial activity that's related to genomics is happening in the United States. And then you see the UK and Germany, France, the usual OECD suspects follow after the United States. But it's predominantly North American story. And this is what those top 15 of those publicly traded companies in genomics are spending in R&D. And the point of showing this slide is, remember there was that huge bubble from 2000 to 2001, but in fact, that's right here. But in fact, the expenditure of these companies continued to go up. Now why is that? Well, it's because these companies were trying to create value by discovering new things, using the tools of genomics, and just because their market value had plummeted didn't mean they could stop spending money. They still didn't have anything to sell until they actually produced something valuable. So they had to continue to spend R&D, and you see that their R&D spend actually continued to increase through 2004. We have not tracked them since 2004, so I cannot tell you what's happened since then. So to summarize that story, we have a field that starts as a pure public works project for creating a public genomic sequence for the good of man and woman that is morphing into something that seems like there's a lot of biotechnology and a lot of private investment going on. So why do we have private investment? Well, just hold that thought because I'm going to come back to it. But that's the story of the Human Genome Project. Now I'm going to switch gears now. I'm going to go back to the field of human genetics, and particularly human medical genetics. And I'm going to make the case that we started with one framework for thinking about medical genetics, and that was rare diseases that were caused by a single broken gene that was inherited in the same pattern that you can see in those 1865 publications by Gregor Mendel. Single broken gene that is either dominant inherited or recessive inherited, or it's on one of the sex chromosomes. And how do you know that? Because you can trace the inheritance pattern through families. So what medical genetics was when I was in medical school was the study of these conditions where you could attribute the phenotype, that is the clinical condition in front of you in the form of disease, to a single broken gene. A Mendelian disorder is what we call that. The task at hand for medical geneticists was to identify those broken genes. Why would you do that? Well, one, as soon as you found a broken gene, you have a way of predicting who's going to get the disease and who isn't. So you have a diagnostic test. That's useful. Two, the real reason that you're really interested in doing that is you don't have a clue what's causing this disease, but as soon as you have a broken gene, you have a link to the protein that it makes, and you have a way into understanding the biology of what is it that's causing Huntington's disease or Alzheimer's disease or Parkinson's disease. You can begin to tease apart the underlying biology by using the fact that there's a single broken gene that gives you the door handle to get into the underlying biology. So what did we do? We would assemble families, and we would try to figure out in those families where's the broken gene. Now, how do you do that? Well, to do that, actually, the way we did that in the 1950s and the 1960s was we would make a guess. We'd say, ooh, okay, so here we've got this disease, sickle cell disease. It's got something to do with blood. Let's look at blood cells. Linus Pauling in 1949 takes hemoglobin, this protein that's in red blood cells, and he puts it on the separation techniques that are hot and new that day, isoelectrofocusing, and he discovers, oh, the proteins are different. People who have sickle cell have this kind of protein, and people who have normal hemoglobin have this. Wow, there's the difference. We can actually see a molecular change. That was the first molecularly defined disease. It took until 1956 to figure out how those protein changes related to the genetic changes, but it turned out that yes, it was a broken gene. It was a single base pair change in the gene that encodes one of those proteins that's involved in forming hemoglobin. So beautiful story, but how did he get there? He made a guess, because what's the blood do? It carries oxygen. What's the protein that carries oxygen? Hemoglobin. Let's see if hemoglobin is broken. Lo and behold, yes. But he had to have a pretty strong prior story to tell. Most of the stuff that we could do that way by making guesses about metabolic pathways and proteins that we already knew about had already happened by the 1980s. So the set of problems that faced us then were a bunch of diseases that are caused by black boxes. My own disease was Alzheimer's disease. And I will tell you, when I was in medical school, there were probably more hypotheses about the causes of Alzheimer's disease than there were medical students in my class. And the idea was, just really any idea was as valid as any other. And one of the techniques that some people began to pursue, including the lab that I happened to work in, was, okay, let's look for families that have Alzheimer's disease, but let's look for the flavor of Alzheimer's disease that's caused by a single broken gene. Now, how would you find that? You would look for families that inherited multiple cases, just like Mendel's peas. So that's what we did, and we discovered families had been reported back into the 1930s. Nobody had really done much with it, but it looked like you could actually find families where Alzheimer's disease looked like Huntington's disease, where everybody knew that it was a Mendelian single broken gene. You assemble those families, and then what are you going to do with that? Well, you need the tools to be able to find a correlation. You know who's got the disease in that family. What you then need is the tools to paint the chromosomes, so you can say, oh, it's this piece of chromosome 4 that's always associated with the people who get Huntington's disease, because they're inheriting that piece of chromosome 4 from the person who got the disease, and they're not inheriting that piece of chromosome 4 from the person who didn't in that same family. That's the technique that you use. What's that called? That's called a genetic linkage map. So the tools for doing that, and the idea of doing this in human beings, actually only appeared in the literature in 1980. So in the 1980s, there is this idea, you know, if we had ways to spray paint, that is to mark the chromosomes, we would have a really powerful tool. That was completely independent of the sequencing stuff that gave rise to the Human Genome Project. But these were techniques for studying human DNA, and what we were trying to do was identify locations where we would find broken genes, and then we would develop, we kind of had faith that we would develop the techniques for being able to go from, there's a gene someplace in this part of this chromosome that's broken, to the techniques that would allow us to derive the DNA sequence and then make a very specific correlation between this mutation and this disease. Because that's really what you're looking for.